It's estimated that antibiotics have saved about 200 million lives since their introduction. But the future of antibiotics doesn't look good if we don't act now.
Why won't antibiotics cure us anymore? This is the University of the Netherlands.
I would like to start by asking a question. What is the most valuable, naturally occurring material? You're probably thinking about gold or diamonds right now. And if you think about the word valuable in terms of monetary worth, you're probably right. But how many lives has gold ever saved? Gold or diamonds are not capable of preventing or curing diseases. In that sense, monetary worth is a rather poor gauge of real value. So, to focus the question, I would ask, what naturally occurring substance has delivered the greatest benefit to human health? Well, as the topic of this talk might suggest, it's antibiotics, at least in my eyes. Let's go back to the year 1900. For people born in Europe in 1900, the average life expectancy was 42.7 years. While today, heart disease and cancer present the leading causes of death, 100 years ago, infectious disease was the biggest killer. At that time, bacterial diseases like cholera, diphtheria, pneumonia, tuberculosis, and typhus were rampant, claiming the lives of young and old alike. This dramatically changed with the widespread introduction of antibiotics in the mid-1940s. As you might know, antibiotics specifically kill bacteria, including those responsible for causing infectious diseases. With the availability of antibiotics, average life expectancy rose sharply in the 20th century. It is estimated that since their introduction, more than 200 million lives have been saved thanks to antibiotics. But how were antibiotics discovered? And what exactly are antibiotics? Well, as the story goes, the Scottish biologist Alexander Fleming discovered the first major class of antibiotics, called the penicillins, in 1928. The best part of the story? He discovered penicillin by accident. Fleming was doing research on bacteria in his lab. Before he left to go on holiday that summer, he placed a bacterium called staphylococci on some Petri dishes so the bacteria could grow in the weeks that he was away from the lab. This is a Petri dish filled with agar. Agar is a gel-like substance that bacteria can eat and grow on. Fleming worked with Petri dishes just like this. After adding bacteria to the agar and waiting some days, the results would generally look like this. Here you can clearly see that the bacteria have grown where they've been streaked out on this agar. However, when he returned from his holiday, Fleming noticed that one Petri dish had become contaminated with a fungus, and that in the area immediately surrounding the fungus, the bacteria had been killed. At that huge, historic, extraordinary moment, Fleming spoke those famous words, "That's funny." In fact, the story of Fleming's chance discovery inspired popular science writer Isaac Asimov to pen his famous quote, "The most exciting phrase to hear in science, the one that heralds new discoveries, is not eureka, but that's funny." Fleming went on to make the all-important connection. He hypothesized that the fungus that had contaminated his experiment was producing and excreting a substance that killed bacteria. That substance, penicillin. As you can see in this Petri dish from my lab, when a tiny drop of pure penicillin is added to the agar, it very clearly prevents bacterial growth, a very nice reproduction of Fleming's experiment. Now, just as any good scientist does, back in 1928, Fleming described his new scientific discovery in a paper that he submitted for publication. However, the impact of his discovery was not immediately clear to the scientific community. In fact, Fleming's discovery floundered in obscurity for more than a decade. In fact, it was the Second World War that led penicillin to be transformed from an academic curiosity into a life-saving medicine. With financial backing from major drug companies and the governments of the United States and Great Britain, the large-scale production of penicillin was realized within 4 years. When the Allies landed on the beaches at Normandy in 1944, they carried with them 1 million doses of penicillin. This gave them a significant advantage in the treatment of battlefield infections against the Germans, who had not been successful in developing effective antibiotics. The success of penicillin ignited the hunt for other naturally occurring antibiotics. As I told you, penicillin was discovered as the product of a fungus. People then began to hunt for other microorganisms that might also produce antibiotics. What followed was the golden age of antibiotic discovery, a 20-year period spanning the mid-1940s to the mid-1960s, where the majority of the antibiotics that we still use today were brought to the clinic. I would like to emphasize that it really is astonishing that so many naturally occurring antibiotics can be used directly as medicines, as is. It's quite remarkable that a compound made by a soil-dwelling fungus or bacteria for its own purposes can also work as a medicine in the human body. It's actually very difficult for medicines to do what they're meant to do in our bodies, and it is for this reason that most other medicines take years to be perfected. In contrast, many of the antibiotics discovered in the Golden Age work extremely well in humans, very much a case of nature delivering us a gift on a silver platter. By the 1970s, the readily abundant, safe, and effective naturally occurring antibiotics had been found. The so-called low-hanging fruit had been plucked. Thanks to a broad arsenal of new antibiotics, bacterial infections were then largely deemed to be under control. As a result, the focus of drug developers began to shift to other disease areas that had comparatively fewer therapeutic options, areas like cancer, heart disease, and neurodegenerative disease. This in turn led to a marked decrease in the discovery of new antibiotics. In fact, over the last 50 years, only 2 new classes of antibiotics have been brought to the clinic. It boggles my mind to think that the vast majority of the antibiotics that we still use today were discovered before I was even born. You might think, so what? It's great that we've been using all these naturally occurring antibiotics for so long. We don't need any new ones because they're working just fine. Well, you would be wrong. The so-called discovery void in antibiotic discovery is actually quite worrisome, particularly in light of an issue you might be familiar with, the emerging threat of antibiotic resistance. Today, life-threatening infections are on the rise as many bacteria have developed the ability to resist the action of antibiotics. This means the antibiotics we have don't kill bacteria they are supposed to anymore. Globally, each year, 700,000 deaths are now attributed to infections caused by drug-resistant bacteria. Notably, many of these infections might have been treatable in the 1960s and 1970s before the bacteria causing the infections developed resistance. To be clear, this means that there are some infections caused by bacteria that were treatable with antibiotics 20 or 30 years ago, but today are very difficult or even impossible to treat. If this carries on, we will have a serious problem. People can die. Healthcare is at risk of moving backwards instead of forwards. Consider also the critically important role that antibiotics play in many routine medical procedures. Thanks to antibiotics, surgeries can be performed without threat of infection. Also, antibiotics provide a protective shield for immune-compromised patients receiving chemotherapy. Without effective antibiotics, such procedures become much more risky and potentially life-threatening. In fact, recent projections suggest that without serious investment and innovation in the field, by the year 2050, antibiotic resistance may claim as many as 10 million lives per year, more deaths than caused by all cancers combined. Where does antibiotic resistance come from? Well, one of the main causes of antibiotic resistance is the process of genetic mutation. Mutations in the genes of bacteria occur spontaneously at a rate of about 1 in 10 million. A bacterial infection represents a population of billions and billions of single-celled bacteria. Due to spontaneous mutation, there will always be a few cells that differ slightly from the others, and in some cases, a mutation can allow a bacterium to resist the effect of an antibiotic. For example, a mutation that changes the structure of a bacterial protein that is the target of an antibiotic can significantly reduce the effect of that antibiotic. Here you see 2 bacterial cells. On the left is a cell with a normal bacterial protein in it, and on the right you see a mutated cell where the target protein has now been changed and has a different shape. The antibiotic here is the yellow triangle, and as you can see, the antibiotic will work on the normal cells. It fits or binds to the normal bacterial protein, allowing the antibiotic to do its job and kill the bacteria. But in the mutated cell, the antibiotic cannot bind its target and therefore doesn't work. This cell is resistant. Such a mutation will initially only impart a small number of bacteria with this advantage, while the vast majority of the other cells remain sensitive to the antibiotic. Bacteria, however, reproduce at an amazing rate, in some cases doubling their population within 20 minutes. This means that an initially small subpopulation of resistant mutated bacteria can, over a period of hours, become the dominant player in an antibiotic-resistant infection. In this animation, you will see that the cells with the blue bacterial protein are disappearing because the antibiotic is killing them. But the cell with the red bacterial protein will not be killed and is therefore able to reproduce. This process of acquired antibiotic resistance and the threat it poses was in fact predicted by Fleming himself in 1945. Fleming then stated, "It is not difficult to make microbes resistant to penicillin by exposing them to concentrations not sufficient to kill them. There is the danger that a man may underdose himself, and by exposing his microbes to non-lethal quantities of the drug, make them resistant." The impact of our enormous use of antibiotics has created precisely the scenario Fleming warned against. Since the mid-20th century, we have injected huge quantities of antibiotics into our environment. Around the world, antibiotics are used not only to treat serious infections in humans, but also on a much larger scale in the agricultural industry, where antibiotics are added to animal feed. Interestingly, antibiotics do not only protect animals from infection, antibiotics also cause animals to grow larger faster. Regular exposure to antibiotics, both inside and outside of hospital settings, provide bacteria with precisely the conditions required for resistance development. If they are exposed to antibiotics frequently, bacteria have more opportunity to get used to them, leading to more resistant cells and antibiotic resistance. So, while our use of antibiotics is vital to maintaining effective healthcare, it has also led us into uncharted territory as a species. We are currently in the midst of a massive real-time biological experiment. How resistance rates will increase in the coming decades is difficult to predict, but the need for action is clear. Okay, so what can we do about this? One approach is to reduce our use of antibiotics. This approach can indeed slow the rate at which resistance appears. It is clear, however, that we cannot completely eliminate our use of antibiotics. Effective antibacterial therapies will continue to be a cornerstone of our medical system. So, we have to look for other solutions. I will discuss 3 such solutions being pursued by my research group. The first solution is enhancing antibiotic molecules. Let me explain what that means. In our research, my team uses organic chemistry to structurally modify naturally occurring antibiotics. This means that we can change or adjust the molecular structure of an antibiotic so that we can enhance its antibacterial properties. Let me give you an example. In the lab, we have found a way to enhance the naturally occurring antibiotic vancomycin so it can overcome resistance. On the left, you see the normal molecule of vancomycin, and on the right, you can see that something has been added. These circles represent new structural features that we can chemically attach to the vancomycin molecule. In doing so, we have discovered new variants of vancomycin that are supercharged, with activities thousands of times higher than normal vancomycin. These new super vancomycins work against vancomycin-resistant bacteria. Let's move on to a second solution. We can also build antibiotics from scratch. In fact, in some cases, this can be easier than trying to isolate an antibiotic from natural sources. For example, some naturally occurring antibiotics are only produced in very low quantities. In other cases, a promising naturally occurring antibiotic might also be toxic to humans. As a solution, we can, in some cases, build these molecules from scratch in the lab, as illustrated here. If an antibiotic is difficult to obtain from nature, chemical synthesis can provide much greater amounts of material. Also, with this approach, we can make new variants with improved properties. As you can see in the image, we can change certain parts of the antibiotic. By doing so, it may be possible to improve antibacterial activity or reduce toxicity. And last but not least, we are working on resistance breakers. This means we are disabling or blocking antibiotic resistance mechanisms themselves. Again, let me give you an example. Some bacteria, called gram-negatives, have an extra outer membrane that can prevent antibiotics from entering the cell. In this way, gram-negative bacteria are inherently resistant to many antibiotics. We are working on taking that outer membrane down as a means of sensitizing them to antibiotics. If we focus on solutions like these, I am optimistic that we can stay ahead of antibiotic resistance. So, as I hope I've made clear in this talk, antibiotics are one of the most valuable natural resources we have ever discovered as a human species. They truly are lifesavers. However, antibiotic resistance is a serious problem, and we are in great danger if we don't act to address it now. The low-hanging fruit in antibiotics was plucked long ago. Thankfully, today, we have new insights and technologies to find new antibiotics. In Leiden, I guide a team of talented researchers who are dedicated to finding new solutions to the problem of antibiotic resistance. Every day in the lab, I keep my ears open in case I hear someone say those famous words, "That's funny." Thank you.